Welcome back. Today I'll explain DNA cloning. So let's assume that this is the strand of DNA that we want to clone. And as we know, DNA is composed of two parts, introns and exons. Introns being the blue part right here, and we're going to have to knock those out. So we're going to end up with a strand of DNA that is all exons, which we call complement DNA or cDNA. Next step, we're going to need a bacterium. And as we know, the bacterial DNA is present in two parts, the bacterial plasmid and the bacterial chromosome. The bacterial plasmid is responsible for conjugation and exotoxin production, while the bacterial chromosome is mainly responsible for cell growth. However, because the plasmid is readily accessible, we can inject our cDNA of choice into the bacterial plasmid. Keep in mind that the bacterial plasmid is actually a part of the bacterial chromosome that has left the chromosome and now is freely floating around the cell. And in a process known as transposition, this bacterial plasmid with the cDNA will be reintegrated back into the bacterial chromosome. So by now we have a bacteria with the cDNA that we want integrated into its chromosomes. And then we're going to put this bacteria into a medium or an agar. And to stimulate its growth, we're going to add antibiotics into this medium. And in response, the bacteria will start growth and multiply. And in doing that, it will also replicate the bacterial chromosome, which has our cDNA. And that is simply the process of cloning.